Hello dear students, welcome to my channel Kaveri Science Tutorials. Today we are going to see an important 5-mark question of meiosis, cell division, meiosis, second type of cell division, meiosis. So meiosis occurs in the reproductive cells and meiosis takes place in two stages, meiosis 1 and meiosis 2. Meiosis 2 is exactly similar to mitosis. We have to see in detail about the meiosis 1. So there are 4 stages of meiosis 1 similar to mitosis: prophase 1, metaphase 1, anaphase 1 and telophase 1. So in prophase 1 of meiosis 1 there are 5 substages: leptotene, zygotene, pachytene, diplotene and dikinesis. So there is a one important 5-mark question in your textbook. Explain the 5 substages of prophase 1 of meiosis 1 in detail with diagram. So this will be beneficial to answer for this question. So the first prophase 1 takes place, there are substages of prophase 1, that is leptotene, first stage. So lepto means thin thread like. So in this stage the chromosomes appear like a thin thread like structure. Lepto means thin thread like, and therefore the chromosomes appear a thin thread like structure and they are clearly visible inside the cell. So next is zygotene. So there is a pairing of homologous chromosomes takes place in case of zygotene, and there is a condensation of chromatin material starts, similar to prophase 1, prophase of mitosis. So condensation you have to write in the second stage, and there is a pairing of zygo means pair. Zygo means pair. There is a pairing between the homologous chromosome. Homologous chromosomes means if these are the two chromosomes. So the genes present on the chromosomes are on the same locus. Homo means same, locus means position. So suppose eye color gene is there, skin color gene is there. So same genes are present on the same arm of the another chromosome. Then it is called as homologous chromosomes. So these types of same genes are present on the same locus, then location or position, that is called as homologous chromosomes. So these two types of these two homologous chromosomes, they pair with each other in the zygotene stage. So it is very important to understand the pachytene stage, because here is a exchange of genetic material, exchange of gene takes place by the formation of a synapses called chiasmata. So chiasmata is the is the point where the non-sister chromatids of the homologous chromosomes cross over each other. So there is a crossing over between the non-sister chromatids of the homologous chromosomes. So we have seen sister chromatid, non-sister chromatids. So these are the these two are sister, but these two are non-sister chromatids. So non-sister chromatids they pair with each other. Non-sister chromatids, they cross over each other. So the crossing over takes place between the non-sister chromatids of the homologous chromosomes, and the point where the crossing over occurs is called as chiasmata, and the point is called as chiasmata, and from that point the genes are exchanged and there is a recombination of gene, and due to that recombination of gene the offsprings are having variations from the parents. Exactly similar नसतात children's त्यांच्या पॅरेंट्सला एक्झॅक्टली सिमिलर नसतात because of this exchange of genes, that is the recombination of genes. So variations, important process of variations in the offsprings occur due to this process of crossing over and the exchange of genes. So that is called as recombination occurs in the pachytene stage of prophase 1 of meiosis 1. So next is diplotene. In diplotene, the crossing over chromosomes, the chiasma appears like a X shape. Chiasmata appears like a X shape. Chromosomes crossing over of the chromosome appears like X shape, and they start to separate by the exchange, after the exchange of genes in diplotene. So this is the characteristic feature of diplotene. So four stages diplotene, and in dikinesis they completely separate from each other with the exchange of chromosomes. So here we have drawn one empty chromosome and one chromosome with shaded part. So this shaded chromosome, so we have to draw here a shaded part like this. Shaded chromosome crossing over. After crossing over this shaded chromosome exchanges, this part is exchanged with this chromosome. Therefore, there is an exchange of chromosome clearly seen in this stage. And one important another step in case of dikinesis is nuclear membrane completely disappears in dikinesis. Nuclear membrane completely disappears in case of dikinesis. So these are the prophase of mitosis. We have to write that a nuclear membrane disappears. Here also we have to write, but in dikinesis. There is a condensation of chromatin material. Here we have to write in zygote. So these are the 5 important stages of prophase 1 of meiosis 1. Again, next is metaphase 1. In metaphase 1, the chromosomes are these exchange chromosomes with exchanges. They are arranged horizontally on the middle plane of the cell, and there is a formation of spindle fiber takes place in metaphase, similar to mitosis. Again, in anaphase, ana means away, they are pulled away from the middle plane and they are collected on the 2 opposite poles of the cell. And in telophase, there is a nuclear means there is a division of nucleus completed, and 2 haploid, after the division of cytoplasm, 2 haploid cells are formed in meiosis 1. So in meiosis 1, there is a important process which makes the difference. There is while dividing the chromosome material from the chromosomes from the middle plane, they are half set of chromosomes moves to the opposite cells of the, so that is the difference. Half set of chromosomes is moved towards the opposite poles of the cell. Therefore, the cells are having half set of chromosome, that is the cells are haploid. So haploid cells are formed in meiosis 1, and meiosis 2 is exactly similar to mitosis. Therefore, haploid cell, again single cell divides, 2 cells are formed. Again another cell divides, 2 cells. In this way, 4 haploid cells are formed at the end of meiosis 2. So this is about the important meiosis division, and these are the important stages. I will again how to remember these stages: leptotin, zygotin, pachytin, diplotin, dikinesis, that is lazy paddy dies. That is the shortcut to remember these stages. So you can take the screenshot of this, and these diagrams are very much important for you, and you can draw this diagram in the examination. So we have completed second division, cell division. In this way, important question, 5 mark question is completed. That will help you in the examination. Thank you.